Basically, I thought this is important information for you guys, and then put it together and let's learn together. So today's presentation is a pretty close to, I try to put those kind of stuff together um, in the way that if you want to dig deeper, you could find the reference. I put a reference besides the slides, so you would be able to see them as well. Yeah, the topic is what Deb said, crop allelopathy and the potential to use for weed control. That's the topic. Oh, my slides not moving quick. So first, let's introduce some kind of a definition. What is allelopathy? Allelopathy is a biological phenomena in which one organisms produce organic compounds, the so-called allelochemicals that affect the growth, survival, and the reproduction of other organisms. So this is a broad definition from biological perspective. For a plant, allelopathy is an interaction between a donor plant and a receptor plant, which can result in either positive, I mean, this positive is from our perspective, not really from plants itself perspective. Positive effects that will suppress weeds and reduce diseases incidence, or negative effects that will be something like autotoxicity. We will be talking about that later in details, or biological invasion. And to some degree, allelochemicals could be treated as, plant growth regulator, herbicides, insecticides, antimicrobial, some of those kind of things that, similar to those conventional sites that use for crop protection purposes. And this is not artificial, but natural. So in the field, it's often difficult to identify allelopathy because there's no way we could separate it from other kind of factors that might be affecting crop plant growth in the vicinity. Because so many factors, we wouldn't be able to differentiate that. For example, the photo on the right side is the sunflower that actually produce this allelopathic to those kind of adjacent plants. And the density are much less. The biomass is smaller under the shed. You might think, well, that's the shed because they probably, block the light. But if you look at in details, and there are some plants around, but near the roots, the plant density is much less. That's the one. And how it worked. The donor plants, through what kind of channel that donor plants would send those allelochemicals to the other recipient plants. The donor plants can release their allelochemicals through, in generally, 4 different ways. The first is the plant manufacture these allelochemicals inside the plant body and execute it from their roots and release them to the soil by roots excavation. And the receiver plants would absorb those kind of allelochemicals, through their roots absorption that would create toxicity to damage the plant or eliminate growth. And a second approach is the leaching by the rain. And those allelochemicals were produced by the donor plant and accumulate on the leaf surface. When the rain start dropping to the ground, and then it will be getting into the soil absorbed by the recipient plant. The third way is the allelochemicals produced by donor plants are volatilized into the air and being taken up by the receiver plant through the stomata. And it doesn't need a whole lot. A small concentration would hurt the plant. And the fourth way is when the plant died or terminated or whatever, they lay on the ground and they as a plant residue, and they start decaying, then the allelochemicals was dispersed into the soil, and then that will be taken up by the plants or affect the germination of the crops, or reduce the plant vigor. Allelochemicals can affect the recipient plant directly by phototoxicity, or it can be affect the recipient plants indirectly through their effects on the soil biota. And when released into soil, allelochemicals degrade to secondary products sometimes. And the bacteria or soil biota play a key role over there, which can either harm the plant, the secondary product, either can harm the plant development or modify the microenvironment around root area, and indirectly affect the recipient plant growth. And the soil biota is very important in transformation of those allelochemicals to secondary products, or it's play a key role in degrading those allelochemicals and make them disappear from the soil. Yeah, I put the references over here, and if you are interested, you could go back to find details of the things in those references. And now let's look at the crops. There are so many plants produce allelochemicals, but this talk we only talking about crops, that popular crops we grow over here, and even not talk. I didn't even talk about those tropical crops. The first one is wheat. Wheat is actually a very allelopathic crop in the agriculture systems. The wheat produces 3 allelochemicals. One is a phenolic acid. It's a hydroxymic acid and a short chain fatty acid. And the research found that wheat residue mulch inhibit ammomereth almost as effective as herbicides. And wheat straw mulch suppress the growth of spinary ammomereth called morning glories and other broadleaf weeds. Water extract of wheat straw adversely affect the germination and growth of several weeds, including field bindweeds, crowfoot grass, purslane, and some forage crop will be affected as well, like oats, corn, sorghum, pear millet, cluster bean, alfalfa, black mustard, and cowpea. So wheat actually is a very strong allelo crop. Wheat crop residue lay on the soil surface. If you have a thick layer of wheat, not really very thick. It's not really go through the physical process. If you put a thick layer of weak residue that block the lights, and which would be similar to some kind of physical blocking thing, and actually wheat residue, if you lay on soil surface not to block the light, exhibit up to 95% control weeds for 30 to 60 days. And once that allelochemicals are released, and then it doesn't have effect anymore. Wheat allelopathic compounds phenolic acid suppression pests and disease as well. And those kind of stuff has been documented thoroughly in the research. And the second crop we're talking here is very familiar. Organic producers already know that 4-rye is very effective to produce allelopathic. What the 4-rye produce is a BX and a phenolic acid and a luteoline glucuranoids found in the 4-rye roots and shoot tissues are phototoxic to many weeds and crops. The BX, we already know that the key allelochemicals that was produced by the 4-rye influence the germination of various dicots and monocots, such as lettuce, tomatoes, barn grasses, etc. And some of the gardeners use this things to prevent other crops, other weeds to grow in their garden. And 4-rye can reduce the weed competitiveness, including wild oats, red roots pigweed, common lamb's quarter, ragweeds, and common purslane. As a cover crop, 4-rye suppress dandelion and and Canada thistle, which has been practiced by organic producers. And when they use as a cover crop residue mulch in soybean, it reduces the weeds pressure and enhances crop yield. A study in Manitoba also showed that 4-rye residue reduces early season broadleaf and grassy weeds population by 44 to 72 and 43 to 88%, respectively. This study actually was done by Dr. Martin's group. And producers in other area, not in Saskatchewan, I saw that they actually, wrote down the 4-rye crop at, flowering stage, and then plant their broadleaf crops, particularly the vegetable crops. And they found that the decay of rye affects the germination of grasses and greatly reduce the weed pressure while the crop is becoming established. That was a very effective way to grow their garden crop and without using chemicals. Next one is the sunflower. Sunflower contain allelochemicals like chlorogenic acid, isochlorogenic acid, scopolamines, etc., that suppress the growth of long list of plants. A recent study that evaluated 8 different varieties of sunflower on weeds control, either by growing this sunflower in mix with weeds or apply the residue at a rate of 6,000 to 1,400, 600 to 1,400 grams per square meter. That's the application rate to the weed crop that has weeds growing in. So what they found is that different sunflower cultivars varied in their allelopathic potentials. Overall, effectiveness of weed suppression is calculated based on each individual varieties. The overall effect would reduce the total weed density by 10 to 87%, and the total weeds biomass by 34 to 81%. So that was a pretty good for some of the crops. Sunflower residue also express their allelopathic potential. They could reduce the total weeds density by 20 to 75%. That's 1/4 to 3/4. And the total weeds biomass reduction by 12 to 67%. And in the meantime, it would increase the wheat yield when they grow in the wheat. Yeah, this experiment was in the wheat field. And oh, they actually found a total of 16 different allelochemicals, and most of them are phenolic acids in different orders. And within these 8 sunflower cultivars, and the cultivars which suppress the weeds contains a high concentration of those allelochemicals. Sunflower can be phytotoxic to some variety of crops like beans, dry beans, millet, sorghum, and corn. Sweet clover and some sweet clover cultivars contain high level of coumarins, which damage the antioxidant system of the recipient plant roots and inhibit root development. High level of coumarin has a very strong inhibitory effect on lamb's quarter, amaranth, barnyard grass, plantain, green foxtail, stinkweed, Canada thistle, dandelion, perennial sow thistle, flaxweeds, and downy broom. The list actually continue going on. What this research found, the research also found that termination at 70% of blooms at that time was often more effective in suppressed weeds than terminate at the bud stage and like earlier stage. And Yukon is a variety with a high coumarin and drought tolerant cultivar is a high coumarin and drought tolerant cultivar and reduce lamb's quarter density by greater than 80% and essentially eliminate the flaxweed. The effect actually continue to the next growing season until the springtime. Kosher density after that sweet clover was reduced by 80%, and white oat biomass was reduced by 30% for the next crop. Application for yellow clover dry powder at a rate of 90 grams per square meter would significantly reduce the number and biomass of weeds. And the next crop is sorghum. Sorghum contains sorghum. It's also hydrophobic benzoquinol and phenolic acids and herrings. Sorghum is a strong allelochemicals that executed, released by the sorghum roots hair. Several studies have found that it inhabits grassy and broadleaf weeds. One study found that applying water extracts of mature sorghum plants reduced weeds density by 35 to 49%. And another research found that applying crushed sorghum mulch decreased the weeds biomass by 25 to 56% in wheat crop and increased the wheat yield by 6 to 17%. There's another study applying about 2 to 6 tons per hectare of sorghum residue reduce the total weeds biomass by around half. Sorghum allelopathy can be applied using sorghum as a cover crop, or you could use it as the intercrop, or include sorghum as a cash crop in rotation to reduce the weeds pressure in your field. Buckwheat is another crop that produce allelochemicals. Uh, the, the allelochemicals produced include flavonoids, phenolic acid, and fatty acids, and alkali, alkaloids. They are very inhibitory to a long list of weeds. The use of buckwheat for weed control has been recommended in Polish agriculture long before allelopathy. That words was, the concept was found, you know, defined. So it was in ancient time. People in Polish already know this buckwheat is can be used for weed control. In lab tests, water extracts from buckwheat they use 10%, like a 10 kilo uh, grams of buckwheats uh, plants with 100 liters of water and soak it for a period of time and use the extract to water the soil and reduce the germination rate of both weeds and crops. But they found that the crop they use survive better. The crops they used for the experiment is the wheat and mustard. They survive better than the wheat. So they basically found, they damaged the weeds more. I guess, that's just give you a concept how that really worked. As a cover crop, buckwheats, and actually some organic farmer already use it as a cover crop. Buckwheats can suppress sow thistle, field bindweeds, rashot, knapweeds, and barnyard grass and quack grass by reducing their emergence and reduce the biomass production and reduce their seed settings and which effectively reduce the seed bank in the soil. And the other field crop includes brassicas. Brassica, brassicas has all have some allelopathic properties as they produce a allelochemical called glucosinolates throughout their plant body, including the leaves and roots. After release, glucosinolate is decomposed into several biologically active compounds, such as isothiocyanate, which play a key role in suppressing weed growth and development. And water extracts of fresh black mustard leaves, stems, flower, roots, and their mixture show inhibitory effects on white oats germination, and the effectiveness increases with the extract concentration. Higher concentration will become more effective. As a part of a cover crop or a companion crop in intercropping, or as a crop in the rotation, brassica can control weeds, and the litter from the brassica can also use as a mulch. Or if it's not enough, you could mix it with other kind of crop residues and mulch the soil. Mustard have a power to suppress many fungal pathogens as well in the soil if tilled into the soil. And we know that some of those crops can be used as a cover crop or intercropping to prevent diseases. That's one of the mechanisms that people use. And the next crop we are talking is the alfalfa autotoxicity. Autotoxicity is the action that a plant releases a allelochemical to inhibit or delay the germination and growth of the same plant species. Basically, they harm their own offspring. It's pretty strange, but it does happen to alfalfa. It helps the plant to gain space for itself to reduce the competition from other, um, you know, plants to compete for water, nutrients, and light. Autotoxicity occurs when the new alfalfa is planted too soon after termination of previous alfalfa crops. The time interval required between the two established alfalfa crop is dependent on soil type, tillage, and soil temperature rainfall. Autotoxicity in sandy soils is more acute, but it's less persistent um, because allelochemicals can be, stay in the soil as a valuable form, not being absorbed by the soil particles in the sandy soil. So they are more efficient, uh, but it will be leached out to the soil, leached out to the soil zone more quickly because the soil texture didn't retain those. Didn't soil moisture was not retained in the sandy soil as the clay soils. So in a clay soil, autotoxicity is less pronounced because some part of the allelochemicals was attached to the soil particles, but it will last longer. They slowly release. They are not easy to be leached out, and the soil conditions favor that favorite that are favorable to microbial growth would reduce the toxins persistency. Aggressive tillage would better mix the soil and dilute the toxins, so the toxicity is greater in non-till field. So if you do have this kind of toxicity thing, and you want your soil to be aerated through tillage, and one is mixing, another one is to help microbiological activity, increase the oxidation speed, and remove those toxins quickly from your soil. So you could just start seeding your next alfalfa crop. The age of alfalfa also affect on the toxicity. So research found that 1 year old or less have less toxins compared to those older plants that has been growing for a few years, a couple of years, more than years. And that's uh, the crop we are talking. And I found that some observations was interesting too. People found that yield of the soybean were lower for several years when growing after flax, and the people start suspect that could be the flax having allelochemicals produced on this plant, but I didn't see the research on that yet. But I also heard several organic farmers talking about very low yield of oats following flax crop. Is that could be allelopathy stuff that was produced by the by the flax, and we don't know yet. The options for allelopathy used for as a weed used allelopathy as a weed control tool from a producer's perspective include this four, and actually we have been talking about that during the presentation. One is the crop rotation and the integration of allelochemicals, allelopathic chemicals crops as a smoother crop that can grow fast with thick canopy that provide additional weed control. And the second one is intercropping and incorporate of this allelopathic crop into as a companion crop that as long as this variety is not affecting, does not suppress the intercrop partner for other one, it should work. And the third one is incorporated as a part of a cover crops, and that would help to reduce the weeds. And in the meantime, some of those allelopathic would also play a key role in control the plant diseases and also used as a mulch. And I guess it's a little bit difficult for field crop producers, but well, some people probably did use that, but it's more often used by those vegetable growers because their system is they hire a lot more people. I mean, and each acre they would produce so much more money, so they could afford to do all those kind of things. And the question might come, I thought I would bring this up, why no commercial products like, currently the chemical kind of product available for conventional producers, why don't they have, develop a commercial product, stored in container that you could grab from the store, use it as, other guys, and which, you know, from some perspectives, it's perfectly suitable for organic productions. The limitation for this, the reason for this include the following. The high cost. Many allelochemicals are very expensive to produce and isolate, regardless how effective that thing is. And if it costs $1,000 for a bottle that could be used for a few acres, and there's no way we could afford to buy those, and nobody would make. And the second part is the public misconception would make the management very difficult. And in general public, many people believe everything that occurs in nature is probably healthy. And this kind of mindset may cause big problems because some of the most toxic compounds known to humans, such as aflatoxin, phenolinsins, and ricin, are all natural. And how do we deal with, particularly like right the ricin was used by terrorists to do the attack. It's very toxic and very effective. So if these things can be used for the cropping systems, we don't know how safe this would be. People mishandle or in contact with could cause huge issues. And the third one is most allelochemicals vanish very quickly. They have very short half-life, uh, last several days to several weeks. Some are even shorter, several hours. And how do you catch that kind of short period, um, you know, to effectively use for the weed control? It's difficult. Well, they vanish quickly actually is something that, preferred. If you could use it on the weeds at the time it affects, and then they quickly kill the weeds and disappear from your system, that would be great. But in most cases, the nature doesn't work that way. And the fourth reason is the inconsistent concentrations, because the production and selection of allelochemicals are highly influenced by the crop itself, its health, growth, age, and vigor, and the environment, temperature, soil, water, nutrient status, and even soil microflora would affect their consistency. So it's pretty hard to produce and produce exactly the same kind of things at this point in time. So those are the limitations at present. But the concept of using a crop allelopathy for weed control is a great concept, and the potential is there for the weed management as a tool for weed management. And I think the future research should be concentrate on the safe use of known allelopathic crop as weed control tools in organic cropping system. Like, figure it out how to use this kind of crops that produce allelopathic and test them on the crops. Is that going to harm the field crop, other field crop for how long? Like, would that allelopathic will last for 4 weeks or 4 months? Something like all those kind of detailed information need to be uh, studied, and continue working on allochemical selectivity and durability. And I mean, find out those new crops that have allochemicals and how that would be able to suppress whatever the weeds in the system and how durability that will be. And third one will be allelopathic chemicals interaction with other crops. That's very important. And just like there are residual effects is also need to be figured out. And the last one is the safe implementation protocols, and develop some kind of uh, protocol guidance for handle those things. Yeah, that's my presentation. Thank you.